It's my pleasure to introduce today's speaker for the MITRE lecture, the Burroughs MITRE lecture. This lectureship was established in 1968 to recognize an NIH intramural scientist with outstanding contributions to biomedical research. And I think today's lecturer, Sriram Subramaniam, is highly deserved of this lectureship. He has been a real leader, a real force in the topic today of cryo-EM electron microscopy. Sriram got his undergraduate training in India in engineering and science, and then a Ph.D. in physical chemistry at Stanford, followed by a postdoctoral fellowship in chemistry and biology at MIT. He is a senior investigator at NCI and is the founding director of the Molecular Microscopy Center, as well as the National Cryo-EM facility, the National Lab at Frederick. He holds visiting appointments at Hopkins and the University of Maryland, and his title today of his talk is the Cryo-EM Revolution. This has been a revolution. This technology has really had great implication from basic science, structural biology, medicinal chemistry. It's really one of these bridging technologies that's had an amazing impact. And if this is a revolution, Sriram is kind of like the Lenin of it or something. I mean, he's really the early thought leader and a very important person who's been pushing the edge in this technology. And so I expect a fascinating talk. So let's welcome our speaker.
Thank you very much, Ned, and thank you, Michael, and many others at the NIH that invited me to give this lecture. If we can have the first slide, please. Yeah, I'd like to actually tell you about the work in my lab in the area of cryo-EM and in related fields. And when I came to the NIH, I have to say it was with a lot of trust that I would do something related to electron microscopy and membrane proteins, and eventually that freedom to do that something really helped us work in many areas that we found very interesting and important. Cryo-EM, as many of you know, and maybe the lights could go down a little bit as we discussed, Antoine. Cryo-EM is now a well-known word. It was labeled as Method of the Year by Nature Methods a couple of years ago, and in the last couple of years, there has been dramatic growth in the field. But I wanted to point out that this is actually not really a new method. Certainly, its impact is seen as being revolutionary, and it is in many respects. But if you go back to a decade ago, as early as 2008, there were structures at near atomic resolution of icosahedral viruses that were being published. Hong Zhao was sort of a pioneer in this area. And over the years, there were many structures at progressively higher resolutions, and you can see this in the chart that captures the growth of the field, not just to obtain high-resolution structures, but resolutions at a variety of at different resolutions ranging from near atomic all the way to medium resolution, where useful biology could be learned. So today I'd like to present a slightly historical lecture of how I and my colleagues came to be in this field, where we started, and how we actually cut a path to be able to exploit these methods to learn to develop mechanistic insights into biological complexes and larger assemblies. Many years ago, when I first was at the NIH, I didn't actually quite know exactly what to do, and Donnie Bliss, who was working in medical illustration at that time, helped me put together this slide, which was a way to think about where the imaging gaps lay in biology and medicine. So in this slide, which has the cliched molecules to man piece covered from the left end, where we think of molecules in terms of Avogadro's number, to individuals whom we don't think of in terms of Avogadro's number, but nevertheless, it gives you a feeling for size, about 27 orders of magnitude. And what's clear looking at this is X-ray and NMR methods have been and continue to be very powerful at the twin ends of the spectrum. And conventional microscopy, light, electron, also has contributed a great deal to our understanding in the middle of the spectrum. But my point, the point that was helpful in guiding the work in the lab, was a recognition of gaps And the one that interested us the most were gaps centered roughly around the size of a virus, and particularly dynamic assemblies that could not be easily addressed by X-ray crystallography because you'd have to capture a particular state, but also where even super-resolution light microscopic methods might not let you get at the kind of chemical questions that we were interested in answering. So the focus of the lab really has been in bridging this gap. And by way of orientation to what actually drew me to this, here's a video shot of a human neutrophil ostensibly chasing a bacterium taken nearly 60 years ago. And this is a 20 kilobyte movie that many of you in the chemotaxis field, cell biologists, are very familiar with this. But to me, what's fascinating about this is while you can make a narrative of what might be going on, such as the neutrophil chasing the bacterium or the bacterium running away, one of them might win. But the point is there's a lot of chemistry that's going on here. There's chemotaxis, there's gradients, there's genes being turned on and off, there's rearrangements of proteins. And I think ultimately we'd like to be able to predict the outcome of dynamic processes like this. And that's a long-term vision. But in the near term, I think one of the important things is to be able to understand all the pieces that are involved in dynamic processes such as this. To this end, we set out an ambitious program to bridge this imaging gap. And it was clear from the early days that we needed tools to go after entities across the spectrum, all the way from proteins, typically with sizes in the range of, say, 10 to 20 nanometers, to viruses, envelope viruses that could never be crystallized, to whole bacterial cells where each cell would be slightly different from its neighbor, all the way to tissue. And there are no simple or single methods that would let you actually determine structures of these entities. And if I were in the extramural world, writing a grant, an R01 grant to determine the atomic structure of a bacterial cell would not be met well. But I could present ideas like that in the intramural program, which really let us take these risky steps towards addressing these problems. And so the work in the lab really was geared towards finding tools that would help us address these gaps by tackling individually structural aspects of each of these entities, all the way from proteins, viruses, and cells to tissue. So today I'd like to essentially provide an outline of this work. I'd like to begin with a very brief introduction to electron crystallography, which is where the origins of this cryo-EM revolution lie. I'd like to touch on some early work we did on bacterial and virus structures, where we developed and used methods in cryo-electron tomography, a technology that is increasingly being seen to be important to study large and complex assemblies. I'd like to touch on something that's slightly off topic from cryo-EM, but nevertheless important in the scheme of things, of how we are using scanning electron microscopic methods to look at very large-scale structures, whole cells, and tissue. And then in the next portion, which really is the primary focus of the talk, I'd like to touch on new developments in cryo-EM and how we are using cryo-EM, and the field across the world is actually using it to get after structures that were previously intractable and to learn mechanism of how these assemblies work. And finally, very briefly, a perspective on where this is all might be headed and how NIH might play a role in disseminating this technology. By way of introduction to imaging with electron microscopy, with transmission electron microscopy, here are two modes in which we collect information in an electron microscope. On the left is the approach that is typically used in cryo-EM and single particle cryo-EM, where in a microscope, electron microscope, that is very much like a light microscope. There's a lens, there's a source of radiation, a specimen. We can collect information in either the diffraction plane or in the imaging plane. If you collect images of individual molecules in different orientations, and you collect many thousands or tens of thousands of these images, these projection images, 2D images, can be combined to generate a 3D picture. And that is the approach that's used in cryo-EM. There's an alternative approach, which I will touch on as well, which is to use tomography, where the idea is very similar to that used in computerized axial tomography, where instead of averaging information from multiple objects, you collect 3D images from a single object by varying the orientation of the specimen relative to the incident electron beam. And that principle of tomography is also very powerful in discerning structures of large entities like whole cells and viruses. the origins of this, as I mentioned before, in terms of using electron microscopes to get at protein structures, really goes back to Richard Henderson and Nigel Unwin in Cambridge, where they demonstrated in the mid-70s that one could use electron microscopes and electron diffraction patterns to determine structures of proteins. And Richard Henderson won the Nobel Prize for cryo-EM last year, along with Joachim Frank and Jacques Dubrochet. And Richard's contribution was to demonstrate that by averaging molecules that were present in a 2D crystal, one could determine an atomic model for bacteriorhodopsin. And this he reported in 1990 after many years of work. And when I joined him at the end of the 1990s, just before coming to the NIH, I was able to work with him and demonstrate that we could indeed get information in electron diffraction space from 2D crystals that went to relatively high resolutions. And that let us determine the structure of this molecule, bacteriorhodopsin, in a variety of conformational states. In particular, we were interested in understanding actually what happens to the protein when it is in the act of translocating a proton. In transporters, this is an important biological question. And this was really the starting point for my own interest in using electrons to derive protein structure. And once at the NIH, we began to apply this to other transporters. And in work we did with Peter Maloney, who was at Hopkins at the time, we began to apply this to 12-helix transporters. And what you see here in the image is on the left-hand side, a typical tubular two-dimensional crystal. And if you zoomed in with the electron microscope, what it really looks like is a collection of molecules organized in a two-dimensional array. And using image processing tools, we can actually determine 3D structures. And in 2002, we demonstrated that we could actually obtain structures of this protein at that time very low resolution, about 6 angstrom resolution. But nevertheless, in combination with work that Ron Kabak had done around that time, we could derive mechanistic insights into the way in which these transporters worked. And over the years, the last decade or so, there's now many, many high-resolution structures. And fundamentally, this mode of action seems to be conserved in transporters in this family. The business of making 2D crystals was a very tedious procedure, and we quickly began to move towards trying to investigate the structures of these membrane proteins in intact cells. And Bob Weiss, who was a sabbatical visitor in the lab at that time, really helped us get started in work on chemotaxis receptors, where we demonstrated that in a variety of bacterial cells, in Colobacter, in E. coli, in Dellavibrio, we could visualize what you see in the left-hand side at the center of the cell, arrays of two-dimensional receptors, chemoreceptor assemblies with their signaling components. And because of the considerable work that had happened in this field in terms of defining the genetic and physiological components, in addition to crystallographic structures of the individual pieces, we felt inspired to begin to use this as a paradigm to learn something about the organization of receptors in intact cells. And this led to a series of papers where we used some of the methods developed in electron crystallography, but applied them to whole cells and derived the mesoscale organization of these receptors and their assemblies in intact cells. And many years ago, a decade ago or so, this was the very first efforts to determine structures of membrane proteins in situ, where we could actually add ligands and look at changes in the organization of the assembly and individual protein structures. But all of this was at relatively low resolution, but it had the value that it let us take on, at that time, what was a considerably more challenging problem, which was to look at these whole cells and look at all the other pieces in it. So this is one example work that Carmen Butan and Jacqueline Mill in the lab sort of pioneered, to look at Dellavibrio cells, where we are now walking into a single Dellavibrio cell, and the level of detail is adequate to actually visualize not just the structure of the whole genome, but each of these single dots are essentially ribosomes. We actually cannot prove they're ribosomes, but they're consistent with being ribosomes. But the fact that you can literally count entities like this is an extremely powerful way to interrogate the architecture of these whole cells. Armed with these tomographic experiences, we then tackled a problem that was important at the NIH and certainly was supported very much by a program, the IATAP program, that Bill Eaton and Michael Gottesman ran. And among other reasons, it was a source of technology support. But very soon, in my lab, we became very much attached to working on this challenging problem. And the central focus of what we did was to take on what seemed at that time an intractable problem, which was to determine the structures of proteins on the surface of viruses capable of infecting cells but where every single variant was slightly differently shaped, different structures, different numbers of these envelope glycoproteins that you see boxed on the right-hand side. So the problem we wanted to solve was to use tomography to actually cut a path to being able to do structures by combining the methods of averaging in 3D with tomography. And we were actually not the first to think about this. Earlier work in the Baumeister lab had led the path to this. And Ken Taylor's group at Florida State had also taken an effort in going in this direction. And once we had an idea of what we needed to do, we began to actually carry out electron tomography of intact viruses. And the method is illustrated here, where we are starting from plunge-frozen specimens of viruses. And in this work and much of the HIV work, we were really enormously helped by a very, very productive collaboration with Jeff Lifson at Frederick and his colleagues, who really taught us and certainly taught me a lot about HIV and the important problems. And the goal of this work was to go from a suspension of HIV viruses in a tube to a map of what the virus looked like in 3D. And this is the unit of data that typically comes out of a single tomogram. It takes about an hour or so to do. But the information here is very noisy because we are collecting these images at very low doses. And the strategy we developed, and this is work that Alberto Bartasaghi in the lab pioneered, was to take tomograms like this and then computationally extract the regions corresponding to these trimeric envelope glycoproteins, each one of which was at very low resolution. But by averaging them and aligning them and classifying them, we then derived the three-dimensional structure of the HIV envelope trimer. In 2008, this was the very first glimpse into what this molecular complex looked like on the surface of the virus. And it really came alive because we had at that time already very powerful structural information from the work that Peter Kwong had done, where he had determined the structures of the individual GP120 protomers, both in combination with molecules like CD4, but also with many other antibodies. And this was very powerful information for us to position these molecules into the density map. And this is an occasion for me to also thank BioWolf and their staff, without whose help we really could not have done any of the work that we've been doing in the last decade. The information that emerges from these tomograms was very useful. It's at low resolution. But because these density maps were at sufficiently high resolution to position structures of the individual complexes into it, it let us develop models for what happened at the surface of these viruses, and in particular, steps that took place in the course of infection. Our interest all along was to be observers at the site of entry between the virus and the T cell, and by bringing essentially pieces of the cell to the virus, we could then begin to visualize conformational changes such as the result that you see here of CD4 binding. And in the course of the last decade, we've done perhaps 100 or so structures of envelope glycoproteins from HIV, from Ebola, from influenza, and that has led to a very powerful database of understanding the dynamics and the antigenicity of these molecules on the surface. Among other things, mechanistically, we were we learned a lot, certainly from being able to develop methods to understand the heterogeneity. And this is a very broad problem in the sense that viruses, envelope viruses like HIV, or influenza or Ebola for that matter, display a rich variety of conformations of different complexes on the surface. And the interest in these infectious viruses also drove methods in the lab of being able to not just describe structures, but also educate ourselves on the plasticity of these envelope glycoproteins to understand which conformations might bind which antibodies less or more tightly. And this early work has led to more recent work on influenza virus, and in a very powerful collaboration with Peter Pelisse and Florian Kramer, we've been extending these methods to try and understand how we can contribute to the development of a universal vaccine for influenza, both in terms of the HA molecule and in terms of the neuraminidase tetramer. And this work is ongoing, and it's been a very exciting aspect of our tomography work. In a similar vein, we've been working with Judy White to work on Ebola, and as many of you know, the ZMapp cocktail has been very effective. And our contribution has been to try and understand where these different antibodies in the ZMapp cocktail bind on the envelope glycoprotein for Ebola, and to be able to discern how these differences might translate into the way in which these viruses enter and infect cells. So far, I've spoken a little bit about electron crystallography and the contribution of tomography to bacterial and virus structures. But I want to digress slightly to talk about the direction we took several years ago to be able to adapt electron-based imaging methods to look at much larger things. One of the challenges with transmission electron microscopy is you cannot look all the way through a large mammalian cell or a collection of cells, simply because transmission electron microscopes, even the ones with the highest voltages, do not really let you look at things that are much thicker than a micron. And mammalian cells can be tens of microns thick. Tissue obviously can be very thick. And I became interested in developing methods to look at these cells, but to go well beyond what we were able to do a decade ago, which is to take thin sections, which is a method that George Palade developed many, many decades ago. So one can take at this canonical picture of a lymphocyte and making contact with a dendritic cell, and if you wanted to know what was at the junction between these two cells, you would then take a thin section. And here's an image taken in my lab. And this junction, which is called the virological synapse, that Tom Hope and Quentin Satentau essentially discovered and characterized in great detail, has a lot of information on cell-cell contacts. And in our case, it has information on how viruses taken up by a dendritic cell or antigen-presenting cells in general get transmitted to T cells. And this is, in HIV biology, this is, of course, a critical problem, where we like to understand early on how these viruses get to T cells and infect them. And to get at this, I had the occasion to know from the work in the semiconductor field that there were technologies based on focused ion beams that would actually let you cut into very large and certainly metallic objects or semiconductor objects. And in a collaboration with FEI company that made these microscopes in the semiconductor world, we began to develop them to use them for biology. And the way these microscopes work is, in addition to having a scanning electron beam to image the surface of objects such as cells or tissue or silicon chips for that matter, they have an additional focused ion beam, which is abrasive and typically made of gallium. These gallium ions essentially, when focused onto the surface, can remove material. And in our very first experiment, we demonstrated using dividing yeast cells that you could focus these gallium ion beams, remove material progressively as you went through the sample, and eventually, as the sample was destroyed, collect these images and combine them to a 3D image. And this was hugely exciting, the fact that we could actually get pictures like this. But a decade or so ago, this was not very exciting, because at that time, you could already get to resolutions like this with confocal microscopy. The resolutions we had at that time were perhaps 200 nanometers, 150 nanometers. But over the years, we are now at resolutions where we are between 5 to 10 nanometers in 3D, in the Z direction. So that, of course, is a very different level of information. But the development of this technology paid rich dividends for the HIV work we were doing. And one problem in particular that we really wanted to understand was the nature of transmission of viruses from a dendritic cell to a T cell. And using these methods, we were able to image in 3D contact zones between dendritic cells and T cells, and recognize that what looked like dendrites in 2D that you see at the left actually are not dendrites, but these are huge walls of membranes that come out of the dendritic cell, and the virological synapse itself is actually nestled deep in this contact zone. And this led to this picture that some of you may recall. This was used in the Research Festival a couple of years ago. Our image of what these dendritic cells look like evolved very quickly to be able to look at them in terms of huge sheets of membranes coming out. And there were further surprises, because we were then able to show that not only was this contact zone covered by sheets of membranes, but the paradigm that we had in our mind for how viruses were transmitted to cells was very different. And what you see here in this image is that as we looked in detail at the synapse, we realized that the T cells that were being infected actually were appearing to infect themselves by reaching into dendritic cells. And this was very controversial at that time, because the idea that somehow the cells that were uninfected would somehow play a role in their own infection was not received without criticism, but we did publish it in PNAS in the end. And ultimately, we went on to show that this was a very common mechanism. And here's an example of synapses between two T cells. The cell in the center is infected with HIV, and the flesh-colored one on the left is uninfected, and we know this because we can see where the viruses are. And yet again, you can see that it appears that the cells that are not infected seem to make tracks towards the cell that's infected. And this is work I want to acknowledge was done by Gavin Murphy, who really pioneered the development of these methods in my lab and unfortunately passed away last year. Yet another example of the use of these methods to unravel the mechanism of HIV transfer came from application to astrocytes. Fetal astrocytes, Avi Nath had just arrived from Hopkins at that time, and they knew how to culture these fetal astrocytes in their lab. And when we began to look at virological synapses between infected T cells and fetal astrocytes, at first it was disappointing because it looked like there was nothing much happening at the junction between these cells. You can see the contact zone seems relatively unoccupied. But when we looked at the same thing in 3D, it became clear that in this contact zone, which developed over the course of a few hours, the astrocytes, which were uninfected, were sending out these filopodial extensions in order that the HIV could surf on them. Walter Mathis at Yale coined this word, and eventually infected the astrocytes. So this idea that uninfected cells play this active role in being infected is now, I think, more accepted than it was at that time. But these insights primarily came from the application of focused ion beam technology. One of the reasons we started to do this was, of course, the application to cancer. And for many years, we worked on melanoma cells to try and understand exactly what it is we can learn from the architecture of these cells as we walk into them. Here's an example of melanoma cell work that Jürgen Heimann started when he was in the lab. And we continue to work in this area actively to try and understand what differences might be between normal cells and transformed cells. But one of the the good things about working in cells and being at the NIH was we began to develop very useful collaborations, and one of them was with Bob Balaban at the NHLBI, and this led to work that we published a couple of years ago, led by Brian Glancy in his lab and Lisa Hartnell in my lab, where we used these methods to visualize the muscle mitochondrial power grid and solve a long-standing problem of how these mitochondria are organized and how energy is conducted. And it led to surprises such as the visualization of single mitochondria that you see on the right-hand side. They don't look anything like what you might find in textbooks. And the fact that we have now a rich sort of deep insight into how these mitochondria might be organized in 3D. And the colors that you see in the lower right animation, each one of them correspond to a single mitochondrion that's connected to others at these junctions. So this is was certainly unexpected. Again, came from the application of these technologies to important problems. The one that we are now very much interested in, and is the beginning, we just launched this collaboration with Luigi Ferrucci, the Institute of Aging, is to begin to use these methods to apply this to a very large cohort of patients whom they study, where they're trying to describe and develop general metrics for aging. And from our point of view, we'd like to contribute to be able to bring structural biology to this problem and to develop methods that might define what it is that changes when people age and what differences might be captured between people of the same age but at different levels of wellness. And this is work that's being done by Brian Caffrey, a graduate student in the lab, Lisa Hartnell, and Marta Gonzalez in Luigi's lab. And here are examples where Brian has developed high-throughput methods to go through very large sections of muscle tissue from healthy volunteers. And this can then be segmented automatically to identify networks of mitochondria. This is completely new information. We want to know how these networks of mitochondria are connected. We can actually go in computationally and extract individual pieces. What you see at the right is a single connected network, probably has many mitochondria in it. But this is information we think might begin to lay the foundations for defining what to look for in this aging cohort. Anyway, these are some examples of how technology for imaging, for tissue imaging, really is likely to be useful looking into the future. For the rest of my talk, I'd like to focus on cryo-EM and how we benefited in many respects from working on these different problems. To be able to take advantage of what has really changed in the last several years. To set the stage for this, if you look at what people studied using cryo-EM, as I mentioned before, it's not a new thing. A decade or so ago, The traditional cryo-EM targets were large entities. Icosiatal viruses. Alistair Stephen at the NIH pioneered this in the study of Hep B, where they demonstrated the very first example of a protein that could be done at 7-angstrom resolution. And this is the kind of thing that people worked on. But what's changed now is from the study of these large entities like viruses and ribosomes, the idea that now with developments in detectors, we can look at these much smaller complexes, much more garden variety examples of cellular proteins, enzymes, and the fact that they're accessible to cryo-EM and might provide information that is beyond what you might get from crystallography. I mean, that is really, in many respects, the excitement in the field. And in our own lab, we worked on a number of these examples at the right end of the spectrum. I'd like to quickly go over some of these examples. For those of you that actually haven't done cryo-EM before, I can tell you on the face of it, it certainly looks easy. And here's an animation of what a typical person might do when they're carrying out cryo-electron microscopy. It starts with a drop of liquid that contains a protein of interest, could be a virus of interest or a cell of interest for that matter. It's delivered on an EM grid that's about 3 millimeters across and blotted with Whatman #1 filter paper that most labs have. And at that point, it leaves behind a thin film of liquid that contains the protein of interest. And when that's plunge frozen in liquid ethane, cooled by liquid nitrogen, the sample becomes vitrified, meaning that the speed of freezing is so quick that it preserves the sample in a glass-like state, and we think this is a near physiological state of these protein complexes. Once it's frozen, the sample stays at liquid nitrogen temperatures and can be delivered into the column of an electron microscope, such as the one shown here, a Titan Krios microscope. And once it's in the microscope, the sample can be imaged, and many of these steps, subsequent steps, are now largely automated, and the unit of information that emerges is a projection image like this, which I mentioned at the very beginning. And each one of these sort of slightly darker things that you see is a projection image of a single protein molecule, in this case, the molecule beta-galactosidase. So if all goes well, we collect thousands of images like this. They can be cut up, processed, and eventually, a few steps later, out comes a density map or a 3D model of the protein of interest at the resolution that the data and the operator are able to get. So this workflow, of course, is not really new. This is what we and many others in the field have been doing for a while. So a decade or so ago, when we began to work on cryo-EM, our focus was very much on metabolic pathways. Our interest was to take this metabolic pathways chart, which all the biochemists of the previous generation had studied in great detail, and have this come alive in 3D. So all of us who've studied metabolic pathways, the Krebs cycle, glycolysis, et cetera, we know what these enzymes are, but what if we could find out what they look like in 3D? And this remains very much an ambition for us to pull together this whole metabolic pathways chart into 3D. And we began to look at metabolites such as acetyl coenzyme A and 2-oxoglutarate, et cetera, and there were some early successes. And that led to a visit from the leadership at the NIH. There's Michael Gottesman, there's Elias Zerhouni, there's Tony Clifford, and a few others. And what they're looking at in this slide is the work that Jacqueline Milne had done at that time, where as a result of studying these pyruvate dehydrogenase complexes, we had begun to put together a mechanism of action of how acetyl-CoA might be synthesized and how the annular gap that the EM images taught us existed in these complexes might be used to shuttle 2 carbons from pyruvate on the outside into the core, where it met up with coenzyme A at the acetyltransferase enzyme, and then eventually made acetyl-CoA. So this was many, many years of work. And the hope that we could do this for smaller molecules was really a dream, but never really realized until revolutionary developments in the field led to better detectors. And for us, certainly in the lab, the key events were 2 papers from UCSF, work from David Agard's lab, from Yifan Chen's lab, that showed that you could actually get to structures at near atomic resolution of membrane proteins and large complexes with these new family of detectors known as the K2 detectors, at that time developed by Gatan. Our interest was to use these detectors and push further. And in the EM field, 3 angstroms was considered much like the sound barrier, and work that Alan Merck in the lab really pioneered led to being able to push these detectors to collect images at progressively higher resolution. And in work that he and Alberto and Sujay Banerjee did, it led to, for us, certainly a breakthrough at that time, was to demonstrate that you could use cryo-electron microscopy to determine protein structures at resolutions approaching 2 angstroms using these direct electron detectors. And among other things, aside from the fact that we had higher resolution, the fact that we could visualize chains of water molecules in a sample of embedded vitreous size was actually particularly exciting, because it tells you that now you're ready to look at the chemistry of how these things work. Another sort of highlight of this work was that the structure we had determined was that of a small molecule bound to a drug PET-G, and we not only could we visualize water molecules, we could look at PET-G, you could look at a water molecule at the junction between PET-G and a glutamic acid residue. So these were exciting times, and once we got this to this stage, it became clear that we needed to demonstrate application to a variety of systems, and we focused on applying this to 3 classes. One was metabolic enzymes of considerable interest to the Cancer Institute and work in the lab. Second, to membrane proteins, and third, to nucleic acid protein complexes. And we systematically went after these and could not have done this without extremely productive collaborations we had both on campus and off campus. The work with, I'd like to describe this in series. The work with dehydrogenases led to this paper in Cell in 2016, where we were able to show that glutamate dehydrogenase could be resolved at very high resolution. And what's interesting here is in this 330 kilodalton enzyme, we could visualize the core of this enzyme at relatively high resolution despite flexibility on the periphery. This is a feature that many proteins will have, and this still, to date, this is among one of the highest resolutions, if not the highest resolution structure reported by single particle cryo-EM. We applied this to other dehydrogenases, but the one that I want to mention is work we did with mutant IDH1, implicated in cancers. And in patients that have a single point mutant in IDH1, that of arginine 132 mutated to a cysteine or other residues, there's a change in metabolism where instead of producing alpha-ketoglutarate from isocitrate, the molecule produces 2-hydroxyglutarate, an oncometabolite. Work at NCATS and actually many other entities have been focusing on finding molecules that specifically target the mutant form but not the wild-type form. And in a very productive collaboration with NCATS, we were able to look at the complex of the molecule that they had developed at NCATS but could not get X-ray structures for with mutant IDH1 and resolve this. And at that time, this was the smallest protein for which near atomic resolution cryo-EM structures could be determined. And the fact that we could locate this molecule immediately opened the door to the next step, which is to understand why it might not have crystallized so easily. And what you see here is that the consequence of binding of this molecule leads to a protein conformational change, and essentially the cryo-EM structure now becomes an assay for binding of this molecule. In addition, we can now compare the location of this binding site to others that have been reported, one from GSK and one from Sanofi, and obviously this is a very exciting area. A second example where we applied this to enzymes is work on P97, and this work was led in the lab by Sujay Banerjee, who I also want to specially acknowledge. Sadly, he passed away last year from CLL. Actually, he was here at the NIH Clinical Center for many of his last months. But Sujay's work led to the structural biology of P97 in the ATP-bound form. More importantly, was at that time the first example where we could resolve a small molecule bound at near atomic resolution. This was published in Science in 2016. And for the first time, we could then show that using cryo-EM, we could derive ligand plots like this, which we were used to from X-ray crystallography, where you could look at the specific interactions of each atom in a drug with its surroundings. In the course of this work, we also recognized something very important, which is why this molecule was so difficult to crystallize in the presence of ATP. And what you see here is because in the cryo-EM work that we did, we were able to resolve simultaneously the presence of multiple conformations. We show that the binding of ATP, the 2 molecules of ATP, first led to a change in one of the ligand nucleotide binding domains, followed by a second set where a second set of changes took place. And it's these kind of conformational changes that take place in these enzymes that really is the power of cryo-EM. The application to membrane proteins is also very, very exciting. And this was enabled essentially solely really with the collaboration we had with Mark Mayer at the NICHD, who's been pioneering the work on glutamate receptors in his lab for a long time. And in work that Joel Meyerson and Sagar Chittori took the lead in my lab, we were able to apply these methods to determine structures of glutamate receptors, GluA2, GluK2, and it led to a number of sort of insights in both the architecture and the gating cycle of these receptors. But I want to highlight in particular two things that are relevant to our own advance in using these methods. One of them being that this was the first instance in our lab where we were able to use native proteins and demonstrate densities for sugar molecules. What you see at the lower right are densities for sugar molecules, N-link carbohydrates. And I think this, the ability to visualize at least the ordered pieces will probably be very important in studies of membrane proteins. So the second insight, unexpected one, was the understanding of the extent of these structural changes that take place with the desensitization of this receptor. And certainly going into this project, we had no expectation that we would uncover these types of very large changes. And again, we're able to do this with cryo-EM because we can classify molecules computationally and determine the presence of multiple conformations. One final example of work where membrane protein conformational changes were analyzed by cryo-EM in the work that we were doing. And this began as a collaboration with Eduardo Perozzo and was triggered by an interesting conundrum in this field because Eduardo had been studying the Core magnesium channel for a long time and had pointed out to us that the structures of this ion channel, of which there were many by crystallography, were essentially identical whether or not magnesium was present in the crystals. And this flew in the face of biochemical work, physiological work that suggested that there were very large changes induced by the presence or the absence of magnesium. And this was a problem that we worked on in the lab, and this was led by Doreen Matthews, a postdoc in the lab, where we systematically went after looking at the structures of Core in the presence and absence of magnesium in a variety of conditions of lipids and detergents. And this was published in Cell in 2016, and it led to a very interesting insight. At first, we were able to show that in the magnesium-bound state, the structure of Core, a relatively small membrane protein by certainly a year ago, this was considered well out of reach for cryo-EM, but a 200 kilodalton membrane protein could be studied by cryo-EM, and the structure in the magnesium-bound state was essentially similar to what we had seen by X-ray crystallography before. But the surprise was that when you take the magnesium away, there's this profound change in symmetry where the protein appears to fall apart. And Doreen showed that this was one of many open states that are populated, essentially an ensemble of states that essentially are populated, very likely because the magnesium binding sites are at the interface between these protomers. So once again, the paradigm of the thinking of these ion channels as being some entities that open and close symmetrically is now clearly no longer the case, and we think that it may well be true that other ion channels also display this type of asymmetric opening mechanisms. The third example of proteins we wanted to actually demonstrate that we could tackle in our lab were nucleic acid protein complexes. And this was a very, very productive collaboration with Dinshaw Patel at Memorial Sloan Kettering Center, where we began to work with him on CRISPR-Cas surveillance complexes. This is a field that this audience needs no introduction to, and it's difficult to keep up with many of the subtypes that are being discovered almost every week, it turns out. But the work in Dinshaw's lab was focused at the time when we began this on a particular class of these complexes, type 1F, and Taiwei Guo in the lab, who was actually a post-bac student, led this work and was able to work with Dinshaw's lab and other people in the lab to actually determine the structure of this complex, both in the presence and absence of bound DNA. And I have to say, this is among the most sort of challenging problems that we had worked on, because this is an asymmetric complex with 9 polypeptides, a guide RNA, and a double-stranded DNA molecule. And we were able to get in work that she and Alberto and Alan worked together to get to these resolutions, 3-angstrom resolutions, where we could visualize the entire complex. Yet again here, dynamics is really what was really interesting. When we compare the binding of the complex. When we compare the structure of the complex before and after bound DNA, there's this dramatic change in pitch that you can appreciate with the structure on the right-hand side. Essentially, the binding of the DNA leads to a reduction in pitch. Essentially, it closes and opens with the binding of DNA. And this ability of these complexes, these ability to essentially be modulated by DNA binding is very likely significant functionally. In addition to being able to look at the result of DNA binding, we were also able to look at a number of inhibitors. These were developed by Alan Davidson in Toronto. And among the many structures that we did of these inhibitors, here's one, ACRF10. And what's mechanistically satisfying is that we can not only determine the structure, but now compare where this sits in relation to the DNA molecule. And in the case of ACRF10, and actually a couple of others, the binding site of these inhibitors is almost exactly where the DNA binds. And once again, these are phage-derived inhibitors. So the interplay between the biology of the bacterium and the biology of the phage, I think it remains really fascinating. A final example of these nucleic acid complexes is work we've recently done, published in Science in 2018. This was led by Sagar Chittori, a postdoc in the lab, and Jingjang Hong, a postdoc in Yawen Bai's lab. And this was sort of an all-CCR collaboration with Alex Kelly's lab, Yawen Bai's lab, and my lab, where we focused over the last year to take on an interesting problem to look at the structure of a protein complex of the centromere. And as some of you will know, the recognition of the centromere is driven by the nucleosome. CENP-A nucleosome actually mediates this recognition with CENP-N binding to it. And one of the challenges in this field is the unique aspect of the CENP-A nucleosome in recognizing CENP-N had been suspected to do with specific residues at the interface, but the structure of CENP-N itself was unknown. That what you see here, the part shown in orange is only about 30 kilodaltons or so. And we were able to use cryo-EM methods to get at the structure of CENP-N and actually position it in the context of the CENP-A nucleosome. And this, among the technical aspects of this work was the ability to actually build a polypeptide fold into this density map despite the low resolution of this interface. But also it was really changed for us the way, it is reflective of the way that cryo-EM is now being used, where without the biochemistry and the cell biology that Yawen Bai's lab and Alex Kelly's lab did, really we would have no idea of how the mechanistic aspects of this complex. Another sort of unique, interesting aspect to this work was as we were doing this and published this, a few days later, work appeared from Carolyn Luger's lab and Andrea Musacchio's lab, where they had the X-ray structure of the CENP-N molecule. And for many months, we were quite nervous about whether we had done this right or wrong. But fortunately, and we could sleep well after this, we could show that the structures indeed match very well. So these are several examples where I think it's hopefully clear that cryo-EM methods can take on relatively challenging examples, things that might be difficult to crystallize, things that might have multiple conformations. But one of the things that we've always worried about is to see if we could actually do better with resolution. And this is work that Alberto Bardesaghi in the lab has been really exploring and pioneering for many years, is whether we can actually make better use of the images that come out of these electron detectors, direct electron detectors, and correct for things that go beyond correcting at the whole image level. This is slightly technical, but in these images, in principle, because each one of these molecules experiences the electron beam in a slightly different environment, if we could figure out how to correct for the local variations, the hope was that we might be able to do slightly better. And Alberto was very successful in this exercise, and this paper has just been accepted a couple of days ago. And we've now been able to advance the resolution much further by making these corrections at the level of single molecular images. And just to give you a feeling for where we've come now since the early work, in 2015, when we reported the 2.2 angstrom resolution structure, you see the top row. You can see that's basically what we were excited about. But now in the bottom, you see this is where we are, where the density map enables delineation essentially of the individual contours of atoms. And sort of a way to illustrate this is sort of in this animation where I'm showing you examples of each type of amino acid where the density is very, very clear and follows the shapes, the individual contours of these non-hydrogen atoms. You can see this also for aromatic residues in particular, where the holes in the middle of these rings of tryptophan, for example, are clear. You can see higher, slightly larger densities for sulfur atoms, and even for negatively charged residues where previously at higher doses we lost density in the carboxylate moiety. Now in many cases we can recover density at these terminal moieties. And finally, for long-chain amino acids such as arginines and lysines, we can still see density all the way out to the end, suggesting they're reasonably well ordered. But the prize really, which we're after, is to be able to do this and locate drug molecules at high resolution. And the improvements in resolution now let us map the binding site in much more detail. And what you see at the left is the visualization of the binding site of PET-G. Each one of these gray contours represents a single atom, a single carbon atom, and there, of course, is the density for the drug, which is a little more flexible, which is a little weaker because it's bound non-covalently. The very last example I want to share with you is a paper literally just accepted provisionally yesterday. It's work on human rhodopsin bound to an inhibitory G protein. And this work actually has very special significance for me because I came to the NIH and actually was given a job to work on rhodopsin. So finally, in some small measure, we can actually show that we can make a contribution to the structure of rhodopsin. And the second reason why this is very significant is this collaboration with Eric Su's lab at the Van Andel Institute grew out of a visit there many months ago, in the middle of last year. And the person who founded this institute was George Vanderwood, who actually was the one who was the director of the NCI, of the CCR, of the intramural program when I came. And I think this is what I told him that we would do when I came to the NIH. So I feel particularly satisfied. And finally, it's interesting to compare structural changes in these seven-helix proteins. So when I first came to the NIH, the work that I had done on bacteriorhodopsin led to discovery of changes which we thought were large at that time, of a few angstroms. But the changes now that take place with these G protein coupled receptors are substantially large. But the interesting mechanistic insight is that the helices that are involved in these changes are remarkably similar. The helix 6 plays an important role, as do some of the other helices. But it's certainly been an important advance to no longer need to depend on 2D crystals to get at these kinds of structures. In the last couple of minutes, I'd like to share my thoughts on how this technology might be disseminated more generally. And for some years now, in discussions I've had with the leadership of the NIH, Francis, as well as many of the institute directors, it was clear that certainly the way that I benefited might be useful for the NIH to sort of expand these efforts. And this is not just an NIH effort. In a piece that I wrote with David Stewart and Nicolas Brescia, who's in Spain, and David is in the UK, there are efforts across the world to make these technologies more easily available, to democratize them essentially. And this led to the founding of centers, certainly in Europe and the UK, and the NIH now is taking a sort of a major leap forward and hopefully announcing the opening of several centers. And in a small way, we actually already have started to do this at the NCI, thanks to the support of Harold Varmus and Doug Lowy, who basically helped, who supported a proposal that I wrote to begin to launch a national facility at the NCI that could begin to disseminate this technology to the extramural world. And right now, we have a single Krios microscope. There will be 2 this summer and perhaps 3 the next year. And there are actually people that do this. This is done under the auspices of Leidos. Ethan Dimitrovsky is the new director of the entire Frederick National Lab program. Dwight oversees some pieces of it, and Ulrich, Thomas, Helen, and Matt, the team that run this. And what's really satisfying is that in the short time that we've been operational, it's been open to the entire extramural establishment in the US. We've had over 20 institutions that have come to use it, over 80 projects, and the need and the use of the facility continues to grow, essentially unabated. So I think that in the years to come, NIH hopefully will invest more in disseminating the technology and make sure that it actually can get to biologists who can actually benefit from this the most. Anyway, to summarize, the cryo-EM revolution is here. It certainly is a golden era in structural biology, but I think its real value is in providing insights into mechanism and especially structural aspects that might actually not be possible by the other very powerful methods, crystallography and NMR. And so looking into the future, clearly it will become an important tool in addition to all the ones that we have. And finally, I do want to say that in the broader sense of the word, although some of the methods we use to interrogate viruses, cells, and tissue don't necessarily use cryo microscopy, it still is useful to view this as a broad discipline where we're using electrons to interrogate 3D structure, be it of proteins or of viruses or cells, and the ability to go into 3D is obviously very powerful. I'd like to end with my deep gratitude to the many lab members who worked with me on all of these projects and really led many of these projects. The current ones are highlighted in blue. I'd like to quickly mention them. Alberto, I've mentioned his name many times, really sort of led the work on the HIV work, on the single particle work, and has been instrumental in many of the technical developments in the lab. Brian Caffrey is a Ph.D. student who's working on the muscle tissue work. Sagar, I mentioned in the context of the nucleosome work. Joseph Darling and Jean-Philippe's work I've not had a chance to mention. They're postdocs in the lab working on various other projects. Veronica Falconieri has been sort of most of the animations I've shown for the cryo-EM work are the ones that she made, so I'm especially grateful to her for working with me and members of the lab to be able to present appealing images of the work that we do. And in the same vein, I also am very grateful to Donnie Bliss, who may be in the audience, who for many years really helped us put a face to these structures, the dendritic cell, the HIV, all of that is really Donnie's work. Joseph Fenney, Andre Florescu are sort of work in the lab and other projects. I mentioned Lisa Hartnell's work with the muscle tissue, Doreen's work on the core. Alan Merck especially, I'd like to acknowledge, has been a brilliant microscopist who's really helped us all the way to really harness the power of the resolution revolution by working at the microscope and also his general insights into how to advance to higher resolution. Janas Ognjanovic is a postdoc in the lab that I have not discussed her work. Erin Tran has been with us for a decade, who's really pushed much of the HIV tomography, and Andrew Wilbur is a postbac who's working with Erin in the lab. The collaborators are too many to go over, but suffice it to say that all the work that I was able to do here was because of my collaboration with Jacqueline Milne, who also happens to be my wife. And when I first came, George Vanderwood said that as consenting adults, we had permission to work together, and I'm happy. I'm really grateful for that opportunity, and also the many colleagues at the NIH that I've mentioned over the years. Finally, this is the present composition of my lab with some of our colleagues at our annual retreat. It's my website, and I'm happy to take any questions. Thank you.
Yeah, so you have any planned applications for this in the diagnostic realm? Where you were talking about melanoma, et cetera, are there any applications pending that you're studying in terms of diagnosis, in terms of some tumor, like tumor margins, et cetera? I know it's a little bit intense and probably goes into a little bit more depth than conventional EM or something like that, but it might be useful in terms of some type of secretions by tumors and margins, et cetera, very cases. Would this be economical to be used in cases such as that in a routine anatomic pathology laboratory?
Yeah, so the question you ask is whether these 3D methods using focused ion beams might be applicable in a clinical setting. And actually, when we began to develop this, that was the primary goal. But we quickly realized that even then, the methods were very, very slow and tedious, and they still are. They're certainly not economical. So I think there needs to be an increase in speed in several orders of magnitude, both at the data collection stage and the data analysis stage. But I think that's the long-term vision, that it adds a new dimension, certainly a new layer of information that does not come out of the conventional pathological assays. That's my hope.
Okay, thank you.
Oh, Sriram, great talk. Can you comment on the future prospects of areas where automation may help the process and reduce the cost? Automation, where can we automate?
The question is there anything left to be automated? Is that what you're asking? Yeah, I think job security is good these days nowadays. I think we're learning as we go. Many of the things we now do are more automated than they were before. But obviously, I think if you look at other technologies, the one thing that seems certain is that things get quicker. Whether they get better, we don't know, but certainly they will be quicker.
There's been some discussion between the crystallographers and the cryo-EM people as to what the definitions of resolution are. Do you care to comment on that a little bit?
Surely you're doing me no favors by asking this question. The crystallographers are used to defining resolution in a mathematical sense. There's diffraction patterns. In the cryo-EM world, resolution has become a very loose measure, and there's a piece that I wrote with Ed Egelman, published a year or so ago, where we actually point out what many of us have known, actually almost everybody in the field knows, which is the way we measure resolution in the EM field is essentially a measure of consistency and not a true measure of resolution. So there's many ways you can correlate signal, you can correlate noise. So these conventional measures, perhaps you're referring to the use of the Fourier shell correlation, is a very primitive measure of resolution. So to me, the reason why the very last piece of data I presented to you, I just said atomic resolution. If you can see the atoms, that's atomic resolution. So at that point, the numbers actually don't matter so much. So I think this is a debate that goes on and on at these garden conferences. But I think if you keep in mind that almost all of these measures are they provide indications of the quality of the map and not to be used as absolute numbers, that's probably a good way to be.
Okay, if there are no further questions, let's thank the speaker. There is a reception